This is the paper. It's the computational design of metallohydrolases. It is a somewhat recent work from David Baker's group. It was submitted actually in late 2024, but it was finally accepted in um, late 2025, and I think finally published in December. So it's essentially just the idea of this de novo design of a new zinc metallohydrolase for a specific substrate that they're interested in. So essentially exactly de novo enzyme design, where they effectively used it as a use case for RF diffusion 2. But as you'll see as I get into the slides, RF diffusion 2 was actually really a very small piece of a very large set of ML models and computational methods in their pipeline for designing these enzymes. So the first question is why these metallohydrolases were hard to design. So the catalysis from these enzymes is dependent on a very precise metal geometry. So you have this essentially zinc T ion that is coordinated by these three imidazole rings for these three histidines. Um, Additionally, the preference is to have these hydrogen bonding groups on uh, other amino acids that essentially stabilize the substrate, and then you need some form of essentially basic amino acid to activate this water to actually initialize the reaction. So it requires both correct metal coordination of the zinc, this water activation from a base, and then this very pre-organized active site. So this is actually from the supporting information of their paper for the entire enzyme design strategy from the computational side of things. I'll briefly go over it, but for the sake of the slides, I'm really only focusing on this, the DFT, the RF diffusion 2, this iterative ligand and PNN and constrained Rosetta relaxation, and then using PLACER as well as a few empirical scoring functions for the final scoring. Um, but as you can really see, they actually started with DFT, and we'll actually go over it here. So first, they actually needed to define the active site specifically for what they were trying to target, which is this very specific coordination site of these imidazole rings and the histidine. Not only that, there are actually four catalytic arrangements that are initially considered and are actually possible because of the different orientations and um, flipped groups of the imidazole. So they actually had to do relatively high level or cheap DFT, really just B3-lip 631 G star level calculations. But they did initial calculations where they actually set up and computed the energies for all four of these different geometries for this rate determining step of the reaction. So this is actually the water, these are the imidazoles, the zinc, and then this is actually the transition state of the reaction for the substrate where you have both oxygens essentially before one is removed. And then these calculations were used to actually provide the initial coordinates of the zinc, the three imidazole rings, and the transition state or the substrate that they used for input essentially into RF diffusion 2. So here's where they actually effectively leveraged RF diffusion 2 is because RF diffusion 2 is this flow matching model that allows for all atom generation instead of just based off this backbone. Instead of having to worry about this very large sampling space of these inverse rotamers and trying to find optimal rotamer optimizations from different positions, they effectively can start from just the enzyme, which is kind of what they defined as the minimal coordination site required from DFT for catalytic function for this given substrate of interest. And then effectively just generate the remainder of the enzyme around this catalytic scaffold or complex. Um, and this is essentially what allowed them to generate these different proposed scaffolds. So for their initial run, they did 2 rounds for the paper. For the initial round and also for the second round, they did, I believe, around 5,100, essentially what they called inferences or full generation of these structures. So they start with a little over 5,000 structures for the first round, just starting from the enzyme and then in painting around it. Yes, so these 5,120. And then these resulting structures were actually first kind of cut out with AlphaFold 2. So they took these initial 5,120 structures and then used protein and PNN to retrieve the sequence essentially from the structure, and then fed these 5,120 sequences into AlphaFold 2. And then they set a PLDT cutoff in the SI they reference, I believe, 0.8 or 0.75, that if it did not align between the essentially design model that they previously had and the AlphaFold2 generated model, that these structures were essentially immediately cut from prediction. Um, and so then they decided to actually further refine them through this iterative sequence design. So they essentially defined two shells. This first shell, which is within about 8 angstrom of the actual catalytic scaffold, and this second shell, which is between 8 and 16 angstrom, I believe, around the catalytic scaffold. And then they essentially used ligand MPNN, which I'll go over briefly next, as well as this actual more empirical scoring function, so Rosetta minimization, and this minor equilibration to try and relax and further essentially improve or find a more stable protein complex around essentially this catalytic scaffold. And so for this, they actually use ligand MPNN in this Rosetta minimization. So essentially ligand MPN is just sequence generation based on conditioning from a given ligand. So for example, you have a protein graph of just the backbone, and then this ligand graph for the actual position of the ligand. And the idea is that you can get some um, sequence essentially from the structure of the protein backbone and the ligand graph. And this was iterated multiple times. I believe in the first round in the SI they referenced, they did 10 iterations or cycles of the remaining sequences through ligand and PNN, followed by Rosetta minimization of the structures, and then referencing again. And then so after all of this, the final designs that maintained the catalytic geometry, so the correct orientation of the imidazoles, a basic residue position to activate the water, and these hydrogen bonds from additional residues to stabilize the transition state were kept and then carried forward again. They then used another model, PLACER, which I believe David Baker's group also developed. It's essentially this gene to produce a ligand conditions protein ligand complex. So essentially a protein backbone in this interaction region, and then some interacting non-protein molecule in a protein sequence. And the idea is that it generates essentially this all atom coordinates of the biomolecular system. They effectively use this as part of their scoring function. So for each of the remaining sequences they had, they actually generated or they ran Placer 40 times with each of them for both the zinc enzyme and also the substrate enzyme complexes. And these scores were kept or tracked for their essentially pocket RMSDs between the design model and these 40 models, and also the histidine or the imidazole RMSDs between the 40. And so what they were specifically looking for by doing this is they want to ensure that the enzyme needs to have a very rigid catalytic geometry. So if you have essentially 40 models from Placer of the sequence, and the geometry of the imidazoles or the catalytic scaffold changes a lot over these 40 models, then it's most likely not an effective enzyme or not a functioning enzyme. And so then they defined effectively the scoring function, which is a combination of the scores from the Placer RMSDs, the Rosetta, which is just an empirical scoring function, and then some scoring function off of AlphaFold2, the PLDT scores, was used to select the initial 96 designs. And even from the original 5,000, with all of these ML models and all of this inference and QM level theory and such, from the first 5,120 sequences or structures that they generated into sequences, only out of the 96, only 86 of them were soluble, and then only 5 enzymes actually had activity over just baseline random noise. And then out of those 5, only one of them had a catalytic efficiency of 16,000 over molar per second, which is still about, I believe, 1/4 or 1/5 the standard activity of these enzymes, actually for wild type enzymes of this type with their substrates. Although they highlight they actually generate these entirely different scaffolds while still having the same activity. Yes. And so this is the comparison of the Placer versus the design. So you can see the highest performing versus the worst performing, there's this very large, essentially, very small RMSD, or very high overlap between all of the predicted models from Placer and the design model, which is why they argue that Placer was useful for ranking these down. And so they actually do a second round where they get much higher improvement. Their reasoning is because in the second round, they specifically know this additional basic residue that is necessary for activity. And so they actually include this basic residue, either glutamate or aspartate, in the thiozyme for all of their generation of the 5,120 new ones, all the way through the pathway. And then once they include this new basic residue in the thiozyme, suddenly they get essentially 85 out of 96 are soluble with 11 enzymes that are now above baseline, with actually one of them, this zeta 2, actually achieving close to, if not standard wild type, so catalytic efficiency of about 53,000 for this novel substrate. And so really the take-home messages from this paper, at least from what I understood from it, is that enzyme design for a novel substrate, even with a very well-known reaction and a well-known family of enzymes like these zinc metallohydrolases, is still a very difficult and intensive process. For this paper specifically, there were a very large number of tools and screenings required computationally, and there didn't seem to be really one or even two models which can do it all. And really from the round 1 or 2, it's that a more fundamental understanding of the chemistry is required for ML models to improve hit rates, because as soon as they added in a more accurate representation of this model for the thiozyme, their hit rate as well as the activity of the actual generated enzymes were much higher.
Yeah. So basically, we still need a better understanding of the catalytic mechanism, right?
Mm,
So that but the fact that they can design an enzyme with a new sub, to me, that is already pretty big success. And
mm.
even the success rate is low, as long as something's working, that's already very impressive.